Okay, if there are no announcements, I will go ahead and introduce Noah. So today I have the honor to introduce one of our round for the departmental seminar, Noah Moore. Noah Moore came to Upstate from Reagan High School, where he graduated in 2014, and then he started at Upstate in 2015, the spring of 2015. I first met him when I was teaching the molecular biology lab. So he was in my section of molecular biology lab. And I realized that he was one of the students who was very motivated and was serious about his experiments. He didn't want anything to go wrong. So I liked him, and I didn't tell him I wanted him to work in my lab, but I think he also thought that he would like to do it. So there was some connection there. So after towards the end of the semester, he approached me and asked me if he would work in our lab. And he graduated that fall, December 2018. And then he immediately started working in our lab in January 2019. So after working for about a year, he had just started knowing how his project is going. And in 2020, the pandemic hit. But at that point, Noah had done a lot of work based on his ethics, lab ethics, and just being motivated about science. So during the pandemic, he spent time thinking about his project well and just reading more and writing. And within that time, he was able to develop scientifically. So I was very impressed. And even lab members were very impressed by his leadership role when we came back to be able to work after the pandemic to join the lab. So he used that time well to reanalyze his data and ask different questions just to see things that he had missed. He has trained several students in the lab. Any student who is in the lab right now, what they know is because Noah has been very instrumental in helping them understand the concepts of malaria and immunology. And he has even trained me. Some of the things since he took over, I haven't been going to the lab a lot, and some things I had started forgetting. Noah has trained me on those, and it is sad that now I won't have anyone to go to since he's graduating. So he has very good skills. So one of the things that motivated him to work in my lab is he wanted to understand immunology. And he told me that he wanted to work on the gut. It is not something I had thought of. And Noah took this project on and started everything by himself. So most of the things he was learning on his own, that enabled him to develop troubleshooting skills, because everything he had to do it on his own. So I've been impressed by just the general learning skills that he has instilled in other students and the environment he has created for us. And it will be an empty nest once he leaves. So I won't take much time to talk about now, because I might start crying. So let's just give him the stage to tell us what he has done for the last year.
Okay, sorry, I couldn't unmute myself there for a second, but thank you, Dr. Apata, for that introduction. So let me close my windows here. So my name is Noah Moore. Like Dr. Apata said, I'm a second-year graduate student here in his immunoparasitology lab, and today I'm going to be talking to you guys about my thesis research that I've been doing for the past 2.5 years. And so my thesis is titled, let me select my laser pointer here, the detrimental effects of malnutrition and Plasmodium trabalii infection on gut integrity and immunity. So before I get started, I want to give you guys a little bit of background. And so my study focuses on malaria malnutrition. So I'm going to go over a little bit of what malaria is first. So essentially, malaria is caused by a Plasmodium parasite, or a protozoan parasite in the Plasmodium genus. There are 4 main strains that can infect humans, and it also occurs in two phases. So there's the sexual phase, which occurs in the mosquito, and the asexual phase, which occurs in humans. And so that's a vector and a host interaction. And then I'm also going to talk about some of the epidemiology associated with malaria, and that includes the symptoms, the outcomes associated with the disease, and also the locations at which it occurs. And so, like I was saying, there's four main strains that can infect humans. And the main one is Plasmodium falciparum. So I say it's the main one because it is most commonly occurring, and it's also the deadliest. And there's also Plasmodium vivax, which is nearly as common but not quite as deadly. And there's also Ovale and Malariae, which are common as well, but they're even far less deadly than falciparum. So our research is really going to focus on mimicking a Plasmodium falciparum infection, which I'll talk about when I get to our model. And over here on the right, I just have an image of the four different strains at different parts of their life cycle. So you can see the ring stage or the immature stage, the schizont stage, so growing and developing, and also the gametocyte stage, which is the adult stage. And the only clear difference I want to point out here is the gametocyte stage, where the Plasmodium falciparum is uniquely different by the fact that it is elongated compared to the other three strains. And now how this occurs in humans. So like I said, this disease is passed on by a vector. In this case, that vector is a mosquito, and more specifically, the female Anopheles mosquito. And when a female Anopheles mosquito is a carrier of the parasite, it can take, or I guess it likes to take up blood meals from humans because that's its food source. And if it is a carrier for the Plasmodium parasite, it can leave behind sporozoites. So when that happens, sporozoites are released into the bloodstream, and the first organ system that they, or I guess the organ that they travel to, is the liver. So the sporozoites will enter those liver cells and they'll continue to grow. And once they grow to a high enough degree, it actually causes the liver cells to rupture, which releases merozoites into the bloodstream. And so this stage of infection is called the asymptomatic stage. And when it's released into the bloodstream, that's when you begin to develop symptoms of malaria, which we consider the symptomatic stage. And it's important to note both of these are occurring at the same time. It's not just like the asymptomatic stage stops and the symptomatic stage begins. But the symptomatic stage, or the blood stage, is where it undergoes asexual reproduction. And so when it undergoes asexual reproduction and that human host is bitten by another female Anopheles mosquito, it can take up parasite as well, and then it will continue to grow and replicate in the female Anopheles mosquito. And at that point, it can pass it on to another human, and the process repeats itself. And like I was saying, some of the epidemiology behind malaria. So it's really important to know the prevalence here. So actually half of the world's population is at risk for infection, and let alone in 2019, there were over 229 million cases with over 400,000 deaths. And what's important about this is 93% of those cases actually occurred in Africa, and actually 94% of the deaths of those cases actually occurred in Africa as well, with 67% of those being children. So children are at really high risk for death from infection. And some of the symptoms that define a general malaria infection are headache, vomiting and nausea, enlargement of the spleen, or also called splenomegaly, pain, fatigue and convulsions in the muscles, joint pain, fever and shivering, and also dry cough. And what's also important to note here is that the different strains also manifest themselves in different ways. So in the case of Plasmodium falciparum, I said it's the most deadly, and the reason for that is because it causes cerebral malaria, which is caused by infected red blood cells sequestering in the brain vasculature, which recruits inflammation and causes a breakdown of the blood-brain barrier. And I'm also going to harp on that a little bit more once I get to my project. So that's something important to keep in mind. And there's also things like vivax, which actually manifest themselves atypically. You can have symptoms such as severe attacks of hiccups, apparently. And then there's also Malariae and Ovale, which generally manifest themselves with these symptoms categorized by febrile paroxysm. So waves of symptomatic and asymptomatic episodes. And what's really important, and to really start beginning on why I wanted to focus on this and the purpose of this project, is this map of malaria and malnutrition. So first I'm going to point out the map over here on the left, which is a map of malaria endemic regions. So the red areas are where malaria is endemic, and the gray areas are areas where it is not endemic. And so I want to point out that the malaria endemic regions are mainly composed of sub-Saharan Africa, Asia, and also Southern America or South America. And along with that, I have this map of malnutrition. So this is a map showing the probability that you would find somebody that is malnourished in any given area. So the darker shades of red are going to illustrate that higher probability. And so we're defining malnutrition here, I guess I should first say, by undernutrition and micronutrient malnutrition, and that's going to be important as well. And similar to the endemic areas of malaria, there's also a high incidence of malnutrition in sub-Saharan Africa, Southern Asia, and also South America. And so this is really important. And before I continue on, I want to give you guys a little bit of background about malnutrition as well. So malnutrition is defined by three broad categories. There's undernutrition, or lack of calories and essentially dietary nutrients that are necessary for survival. There's nutrient malnutrition, which is a lack of micronutrients such as zinc, iron, and protein, and also overweight malnutrition. And we're specifically focused on undernutrition and nutrient malnutrition. And these actually affect approximately 2.4 billion people per year. And just like malaria predominantly affects children under the age of 5, these forms of malnutrition also drastically affect children under the age of 5, causing 45% of deaths in those children. And like I said, there's also high occurrence of these kinds of malnutrition in developing countries. And so while that's some good background information, what I want to point out is this figure over here on the right, which is the general effects of malnutrition when considering undernutrition and nutrient malnutrition. And there's really two main ones here. The first one being impaired gut integrity and immunity, and also decreased immunity and resistance to infection. And there's three specific nutrient deficiencies that I want to point out, and this is going to make a little bit more sense once I get to our model. But this first map is showing the global proportions of zinc deficiency. So zinc is a really important nutrient involved in white blood cell proliferation and the development of a robust and proper immune response to infection. So when you have deficiencies in zinc, you're going to have a less robust immune response. And again, I want to point out that the highest proportion of individuals affected by zinc deficiency is in sub-Saharan Africa, Southern Asia, and again, South America. Next, I have a map of iron deficiency causing anemia. So this is more so a map of anemia caused by iron deficiency. And again, I want to point out that the largest degree of anemia due to iron deficiency is also occurring in sub-Saharan Africa, Asia, and also South America. And lastly, a map showing the incidence of protein energy malnutrition deaths per 1,000 children under the age of 5. So this is also called PEM, which is due to a lack of protein. And similarly to zinc and iron deficiencies, the highest proportion, or I guess in this case, the highest number of individuals affected is again in sub-Saharan Africa. So this is really important because what we want to hypothesize here, based on what we know now about malnutrition and malaria infection, is that they might be exacerbating the effects of one another. And in order to explain that further, I have two images here. And I first want to draw your attention to the image on the left, which is a villi in the gut. And so what's believed to happen during malaria infection in the gut is that those infected red blood cells can sequester or adhere to the epithelial wall on the inside of the villi. And when that happens, you have immune cells that are recruited to that location to try to fight or kill those infected red blood cells. And by doing so, they release inflammatory cytokines. And you have to think, if you have a highly inflammatory environment in a localized tissue, there's going to be breakdown of any kind of barrier there. And this is what's believed to happen, is that that barrier in the gut epithelium is broken down. And when that happens, you can have dysbiosis of microbial communities, and you can also have bacterial translocation into the bloodstream, which can affect other organ systems. So that's what we hypothesize is happening in the gut during malaria infection. And over here on the right, I have what is suspected to happen during a zinc deficient condition in the gut as well. And before I explain this, I want to point out that the same effect I'm going to talk about here is also seen during iron and protein deficiencies as well. But first, I'll focus on the healthy condition. So when you have enough zinc in your diet, your gut barrier is very well maintained. And specifically, I want to point out these black bars that are illustrating the tight junction proteins. So those anchor the epithelial cells together, and the expression of those is enough to keep the epithelial cells, of course, anchored together so you don't have any kind of dysbiosis or bacterial translocation happening. However, under zinc deficient conditions, and again, I want to point out that similar results have been seen during iron and protein deficiencies, it's believed that there's a breakdown or a disarrangement of these tight junction proteins. And when that happens, you have loosening of your epithelial barrier in your gut, and your potentially pathogenic bacteria can pass through and cause barrier dysfunction and ultimately recruit inflammation. So with these two things in mind, again, I want to reiterate that we're hypothesizing, well, malaria infection must exacerbate the effects of malnutrition in the gut. And in order to do that, we used a mouse model, or as we call it, a murine model. And these, or I guess the mice we used were standard C57BL6 mice. So you can really find these in any lab across the country. They're the standard for rodent research. And so what we did in order to study the two effects is we fed first the mice a control diet or a malnourished diet. And so the malnourished diet is deficient in zinc and iron, and it has 3% protein content, versus the control diet, which has all the necessary nutrients. In this case, it also has iron and zinc, and it has 17% protein content. So we fed the mice either of these diet for 4 to 5 weeks to induce moderate malnutrition. And at that point, we infected them with Plasmodium chabauti. And so Plasmodium chabauti is the rodent strain of malaria that mimics a Plasmodium falciparum infection in humans. So the most commonly, or the most common Plasmodium parasite, and also the deadliest Plasmodium parasite in humans. But what's important is that we infected them with a concentration of 1 × 10⁵ infected red blood cells. So that's not enough to cause severe infection, but is enough to cause chronic infection and induce a pretty drastic immune response. And so that brings me to the next point, which is the day 9 time point. So we waited for 9 days after infection to begin to analyze the tissues of these mice. And the reason for that is that day 9, or the day 9 time point, is when the parasite is going to be at its highest level in these mice, which is going to produce the most robust immune response. And you're also going to see the biggest differences in various tissues. And so at that point, we did our gut analysis. But first, I want to talk about some of the pathologies associated with malaria infection and malnutrition in our mice. So first, I want to draw your attention to panel A, which is indicating the percent parasitemia. So the percent parasitemia, we got this information from taking a tail snip of the mice and taking their blood and staining the red blood cells. And so what this is a ratio of the amount of infected red blood cells with malaria over the total number of red blood cells that we can count. And what's interesting here, so the circle is showing the control mice that are infected, and the open circle is the malnourished mice that are infected. And what's actually important is the control mice during infection actually had a higher percent parasitemia than the malnourished mice. So the next thing we did is we looked at temperature change. And so we took the temperatures at days 0 through 9 post-infection via rectal thermometer. And again, the closed circles are the control mice, the open circles are the malnourished mice during infection. And what we actually saw is the control mice tended to have a larger decrease in their body temperature compared to the malnourished mice. And one thing I want to point out about this is that during infections, the body temperature of a mouse will actually decrease because they can't produce fever in response to infection. So this is how they try to kill that pathogen, by lowering their body temperature. And so this begins to make a little bit of sense when you think about it in terms of the parasitemia. If the control mice had higher percent parasitemia, you would have to imagine that they're going to also have lower body temperature because they have more parasite present, causing more symptoms of disease. And the last thing we looked at in terms of pathologies is the percent total change in body weight of these mice at days 0 through 9 post-infection. So I first want to draw your attention to the squares. So the closed square is the control mice without infection. The open square is the malnourished mice without infection. So we do actually see that the diet caused a significant decrease in weight compared to the control mice without infection. However, when we did infect the mice, again, looking at the closed circle and the open circle, there was no significant, or there was no really true significant difference between the two groups during infection. However, what's really important and interesting about this data point is looking at the control mice without infection compared to the control mice with infection. They have a larger decrease in weight when comparing infection to no infection versus the malnourished mice, the open square versus the open circle. There's not that drastic decrease in weight when infection is introduced. So this is actually indicating that the control mice are experiencing the greatest pathologies associated with the malaria disease. And so that brought us to our first analysis on the gut. So we first wanted to do a really broad scale macroscopic analysis on the gut to really see if there's anything going on. And so this involved taking those mice at day 9 post-infection. We took out their small intestines, their large intestines, and the cecum, and we did a macroscopic analysis, which was essentially just us looking at the tissue, and we also took the links of the tissue, and that's going to be a really important piece of data. And so these are some representative images of each group of mice. So I first want to draw your attention to the control uninfected versus the malnourished uninfected. So when looking at the control mice, you can see there's a lot of redness to the tissue and everything looks pretty healthy. But when we compare that to the malnourished uninfected, you can begin to see there's almost what appears to be a yellowing of the tissue, and it also appears they're eating less. However, now looking at the control infected versus the malnourished infected, the control infected mice, there is a loss of redness compared to the uninfected, so it indicated that the malaria infection is impacting these in some way. However, what's more important here is the malnourished mice during infection. There's really a large increase in what we call the yellowing of the tissue, and even more importantly, so I'm to the left of my laser pointer, you can see that the tissue is almost transparent at different parts. So this doesn't really give us any conclusive results, but it starts to indicate that something is going on in the presence of malnutrition and infection. And the next step in determining, well, what's going on here was taking the links of each respective tissue. So the small intestine, the large intestine, and the cecum. And so this is a really important piece of data, although it might seem kind of really basic. It's really important because shortening of the intestines or any kind of tissue is actually associated with an increased inflammatory environment. So when you have increased inflammation in your intestines, there's, like I said, that breakdown of the tissue, which results in fibrosis and inflexibility, resulting in shortening. And what's important to see here, first I'm going to again point out the control uninfected versus the malnourished uninfected. And what we saw is that with the malnourished diet alone, there tends to be a decrease in the total small intestine length compared to the control mice. And again, that's not significant. It just tends to decrease during the diet. However, more importantly, when we compare now malnutrition and infection, we see a significant decrease in the total length of the small intestine compared to the control mice during infection. So this is really indicating that malaria infection is exacerbating the effects of malnutrition, in the small intestine. And again, I want to reiterate that this could potentially be indicative of an inflammatory environment, which will be really important later. And so we looked at the same piece of data in the large intestine, and we saw some similar trends, but nothing of true significance. We just see that general decrease during malnutrition, and also malnutrition and malaria infection. And we looked at the lengths of the cecum, and we didn't really see anything of interest there. And so I pointed out that shortening of the intestines is associated with inflammation and potentially breakdown of that epithelial barrier. But before I get ahead of myself, I want to point out what we did next, which is a analysis with this molecule called dextrin. So under normal conditions, when the gut epithelial barrier is intact, the dextrin molecule can't pass through and get into the bloodstream. But when you do have breakdown of the epithelial barrier in your gut, the dextrin molecule can leak through and get into the bloodstream. And this is really important because this molecule of dextrin was labeled with a fluorescent marker called FITC. So we could then take out the blood of these mice at day 9 post-infection and measure the differences in dextrin concentration in their blood serum, which would indicate different levels of gut integrity. And of course, we measured that on a spectrophotometer. And that brings me to my next piece of data here, is the concentration of FITC dextrin in the different groups of mice. And again, higher concentrations of this are going to be indicative of a loss of gut integrity. And again, I first want to point out the control uninfected versus the moderately or the malnourished uninfected. And so what we see here, although again not significant, is that the malnourished uninfected mice tend to have a higher concentration of dextrin in their blood versus the control uninfected, which again, that indicates some kind of loss of gut integrity. And more importantly, looking at the control mice and malnourished mice during infection now, you can see that there is an increase in concentrations of dextrin in both of their blood serum during infection. And more importantly, this effect is increased or exacerbated in the malnourished mice. So they have a really high concentration of dextrin in their blood serum compared to the control infected mice. So this was a really exciting piece of data to get because it's really starting to show the loss of integrity during malaria and malnutrition. And what we did to further investigate this was H&E staining. So many of you should know that H&E staining is used for microscopic analysis now. So again, we're taking these mice at day 9 post-infection. We took out their small intestine, fixed them in PFA, and stained them with H&E to really look at differences in that epithelial layer. And this is what we got. So these are representative images of our H&E staining in the small intestine. So first, I want to draw your attention to the control uninfected mice, and the black arrows are pointing to epithelial cells. And what I want to point out here pretty simply is there is a good degree of organization with these epithelial cells. You can see that there aren't any gaps between the cells themselves. However, during malnutrition and the absence of infection, you can see that we begin to see some disorganization in the tissues. There is some white space in the actual epithelial cells. And next, looking at the control mice during infection. So these are a little bit different from the control uninfected by the fact that we begin to see some spaces forming in between the epithelial cells. There's some disorganization at the villus layer. And more importantly, again, the circumstance of malnutrition and infection. And what we see here is a large degree of disorganization. So these epithelial cells are really largely disorganized. There's a breakdown of the tissue so much that some of the epithelial layer is actually scattered into the gut lumen. And more importantly, actually pointing at this epithelial cell here, it actually appears it's been destroyed. So this was really promising and really began to show the effects of malaria malnutrition on gut integrity and a breakdown of the epithelial layer. And our next task was really finding out, well, why is that happening? And a way we could answer that was with flow cytometry. So what we did is we took these mice again at day 9 post-infection. We took out their small intestines, their large intestines, and cecum, and we processed them with FACS analysis. And that would enable us to then look at different types of cells that could be causing inflammation and that breakdown of the gut integrity. And so what I have here is a image of what this analysis is. So this FACS analysis or flow cytometry. So this is really great to show you guys because this simplifies my my 20-hour experiments down into one image that I can explain to you relatively quickly. So the first process in this is taking those tissues and making a single cell suspension. So homogenizing them and suspending them in media. And by doing that, we get single cells, and we can label those cells. Again, this is a homogeneous mixture of different cell types, and we can label the specific ones we're interested in with antibodies, and those antibodies have a fluorescent molecule that can be read by the flow cytometer. So we put those cells that were stained with antibodies through the flow cytometer, and it shines a laser on those fluorescent molecules, and depending on if it fluoresces or not, it's detected by these detection emitters. And that can be processed on a computer, and we can begin to see differences in proportions and also cell numbers. And that brings me to my first piece of data that was associated with our FACS analysis. So what I did first is I looked at early CD11B cells. So these are early progenitors that could develop into macrophages. I looked at myeloid cells, which are CD11B, CD11C positive, and I looked at early dendritic cells that are CD11C positive. And I first want to point out over here on the left, the early macrophages or the CD11B positive cells without infection. So you can see that there's relatively low numbers of these cells in both groups without infection. However, when we infect the mice, so the control infected mice and the malnourished infected mice, there's a big increase in CD11B positive cells. And what's kind of important to note here is that the control mice tended to have more CD11B positive cells than the malnourished mice, though again, I want to emphasize that that was not a significant difference, but we do see a trend towards a decrease during malnutrition. And like I said, we also looked at the CD11B, CD11C myeloid cells. And again, first looking at the uninfected mice, there weren't many of these cells that abundant in the small intestine. But again, during infection, either in the malnourished mice or the control mice, they are increased with a slight increase in the control mice, though not significant. And lastly, the CD11C early dendritic cells, again, not very many of them without infection, but during infection, the control mice tended to have more than the malnourished mice, though not significant. So we looked at this data in the small intestine. We also looked at the same cell types in the large intestine. And again, pointing out without infection, there aren't many of the CD11B positive cells. When we infect them, there is an increase in the control mice of those CD11B positive cells. They're not significant. And again, we looked at the myeloid cells without infection, not many of these cells present, but when we do infect the mice with malaria, there's an increase in total cell numbers of myeloid cells in the large intestine. And lastly, the CD11C positive dendritic cells without infection, not many present, but during infection, there's a significant increase in the control mice versus the malnourished mice. So that is the data in the cecum, or sorry, that was the data in the large intestine. The next thing we did was we looked at the same cell types in the cecum. And again, I just want to say that we saw pretty similar trends here, though nothing really significant. Our error was kind of high, so it's really hard to conclude any differences here. So this posed a major roadblock in my research. I kind of had to wrap my head around a little bit, and luckily COVID gave me time to sit down and think about it. So what I want to point out here is this data from the small intestine. So that increase during in these cell types in the control and infected mice. So the CD11B positive, CD11B, CD11C positive, and also CD11C positive cells were increased, not significantly, but tended to increase in the small intestine during infection. And what's important to note, is that those early cells eventually cause downstream inflammation. And how that really doesn't make sense, or why it was kind of frustrating to see this, is because we saw the most amount of inflammation and breakdown of gut integrity in the malnourished mice, not in the control infected mice. So this didn't quite explain our H&E staining data, nor did it explain our dextrin analysis. So in order to bring forth what we did next, I need to give you guys a little bit of background. So this is not my data. This is a Western blot from the Journal of Experimental Medicine in 2002. And to give you guys a little bit of background here, I want to point out that IL-12 and IL-18 are actually shown to be really important cytokines during malaria infection that actually impact the severity of disease. And so what they did is they cultured macrophages, so CD11B positive cells with IL-12 and IL-18, and they studied interferon gamma production or expression, which is a really important mediator of gut integrity and also inflammation during infection. And what they actually found is that dependent on concentration, macrophages, when they are cultured with IL-12 and IL-18, produce significantly more interferon gamma. So that's an important piece to keep in the back of your head. During malaria infection, macrophages are secreting a lot of interferon gamma. And what's also important is this image here. So this is an image of the gut, and this is what you could argue a villi, or a dip between the epithelial cells. But what I want to point out is this area right here. So at the site of epithelial injury, in the case that we saw with our H&E staining data, macrophages are recruited to that location, and they are charged with fighting bacterial translocation and also restoring this barrier integrity. And one of the major cytokines they produce to do this is interferon gamma. So what I'm really getting at here is that macrophages secrete a lot of interferon gamma during malaria infection, and they also secrete a lot of interferon gamma during breakdown of the epithelial barrier in the gut. So things begin to make a little bit more sense when you think about it this way. So the last image I want to talk about here is over here on the left. And what I specifically want you to know about this image is these macrophages here. So we can see that when they are activated, so this is an MHC2 molecule, which I will explain next, that's going to be really important as well. They can interact with T cells, and those macrophages actually have an interferon gamma receptor, which can influence them towards an inflammatory phenotype. So you have to think about this in kind of a positive feedback loop. If macrophages can secrete interferon gamma, and they can also have a receptor for interferon gamma, there is going to be a positive feedback loop that's going to produce a lot of inflammation via production of interferon gamma. And that ultimately results in a lot of inflammation and also fibrosis, like I pointed out earlier with the shortening of the small intestine. So when we think about things in this way, it begins to explain what we saw here a little bit better. But I can't just say that this explains our data. We then sought to actually investigate if this was the cause behind the increased damage in the malnourished infected mice's intestines. And in order to do that, again, we used FACS analysis, so using flow cytometry to look at proportions and cells of interest, in this case in the small intestine, the large intestine, and cecum of our day 9 mice. And another piece of information here, the last piece I promised before I get to the good parts of the information. What happens during any kind of bacterial infection is that, or in the case of this also breakdown of the epithelial barrier, is that macrophages take up antigen, and they can process that antigen and present it on an MHC2 molecule, which I kind of foreshadowed earlier, and present that to a CD4 T cell, which would then produce an adaptive immune response. And the important part about this is that macrophages, when they express MHC2, they are considered activated. And at that point of activation, they also begin to secrete cytokines. They're not shown here, cytokines like interferon gamma. So we wanted to look at this activation phenotype before we really got into cytokine production. And that brings me to this really important and really exciting next piece of data here, is the activation phenotype or the antigen presentation phenotype of these macrophages in the small intestine. So we looked at the proportion of CD11B positive, MHC2 positive macrophages in the small intestine, and also the number of those same cells. And what we begin to see here, well, I guess first I'll point out, in the absence of infection, there's not really any proportion or increased number of these cells. But when we infect either the control mice or the malnourished mice with Plasmodium schaubouti, you can see that now things begin to tip the other way. These mice tend to have an increased proportion of macrophages expressing MHC2. Again, although not significant, there is a trend towards an increase here. And there's also a trend towards an increase in the number of these cells as well. So the malnourished mice tend to have higher numbers of antigen presenting macrophages, which indicates that activated phenotype. And again, this is occurring in the small intestine. And similarly to our last experiments, we looked at this in the large intestine as well. And without infection, again, the proportion of activated macrophages are relatively low. However, when we infected the mice, especially in the case of the malnourished mice, there is a really significant increase in the proportion of activated macrophages in the large intestine when comparing those to the control infected mice. And again, we also looked at the number of these cells as well, and we saw the same trend. Without infection, there's not many activated macrophages. With infection, there's an increase, and there's a trend towards a bigger increase in the malnourished mice as well in the large intestine. And lastly, again, of course, we looked at the cecum, and again, seeing much of the same trends. So without infection, lower proportion of these cells. With infection, there's an increased proportion with the trends towards an increase during malnutrition compared to the control mice during infection. So the proportions, and again, the numbers are increased during malnutrition and infection, although not significantly. So that was really exciting because that finally got us on the right track with thinking about this project. And really to bring this all home, is we wanted to actually look at that interferon gamma production that I pointed out earlier, which is responsible for gut barrier integrity and also for malaria infection. So that's exactly what we did. We took the mice at day 9 post-infection. We took out their small intestines. We processed them with the FACS analysis, except what we did this time, which is unique and different, is we did an intracellular cytokine staining. So by doing so, we're able to look at the amount of interferon gamma. And what this process entails is treating cells with a drug or stimulant. So again, we took these cells, we cultured them with PMA and ionomycin, which is a stimulant, and we put them in an incubator. And then we also fixed and permeabilized them. So we're fixing the membrane so we can then poke holes in that membrane to look at the cytokines. So we fixed them and we, like I said, we poked holes in them. And then by doing so, we can stain those cytokines that are produced on the inside with antibodies. So again, that's what we did next is we stained those cytokines, in this case, interferon gamma on the inside with fluorescent antibodies, which we then could process similarly to our surface stained cells that I pointed out earlier. And this was really exciting to see. This is data from the small intestine. So again, we looked at macrophages, so CD11B positive, F480 positive macrophages that are secreting that inflammatory interferon gamma, so the proportion and also the number. And this is in the small intestine. And again, I first want to highlight the absence of infection. So the malnourished, or sorry, the control uninfected and the malnourished uninfected tend to have lower proportions of macrophages secreting interferon gamma. But when infection is introduced, there's an increased proportion of macrophages secreting interferon gamma during malnutrition compared to the control mice. And again, that's not significant. It's just similar to all the trends we've really seen here is it's a trend towards an increase. And I think what brings things all together here, brings it all home really nicely, is this figure here. So this is the number of macrophages secreting interferon gamma. And this is more important because that actually shows the amount of cells in that particular tissue So this is more dependent on the amount of cells we collected. This is more representative of the actual differences between cell number. And again, the uninfected mice had a really low number of macrophages secreting interferon gamma. However, really exciting here is the macrophages. Well, sorry, I'm getting ahead of myself. The malnourished mice during infection had significantly more macrophages secreting interferon gamma than the control infected mice. So this begins to really explain that inflammation we saw earlier during malnutrition and infection and the breakdown of that gut epithelial barrier, and also increased gut integrity. And so I want you guys to take away really two simple conclusions from this that I'm showing here. We think that malaria infection is exacerbating the effects of moderate malnutrition on gut integrity and also morphology. And secondly, we think that malaria infection also increases the innate immune response during moderate malnutrition, which results in more inflammation. And lastly, I drew up a little graphical abstract kind of showing that as well. So this is the epithelial damage that we think is occurring during Plasmodium chabauti infection, either in the control mice or the malnourished mice. And so what we think is happening is that in the submucosa of the malnourished mice during infection, there is an increased number of macrophages, so these purple cells here, secreting these red dots, interferon gamma. And by doing so, they're actually, what we think is happening is they're actually causing increased damage to the gut epithelium versus through that positive feedback loop that I mentioned earlier. This is compared to the well-nourished mice. So the control mice during infection, we're suggesting that they have less macrophages present and therefore less interferon gamma production, which is allowing that gut epithelial barrier to remain intact. And some future directions for this project. You remember I said earlier that it's hypothesized that the parasite can sequester in the villi. That's not actually been shown. It's just that's what the current consensus is on what people think is happening. And so we actually want to investigate parasite sequestration in the villi to see if that would really explain what's going on here. And we can do that through techniques such as immunofluorescence. And also really important is we want to look at tight junction integrity. You remember I said tight junctions are really important for keeping that epithelial barrier tight and make sure there's no dysbiosis and translocation occurring. So we want to investigate the expression of those with RT-PCR, and that can be tight junction proteins such as zonulin and occludins. And also we want to look at the resulting microbial community dysbiosis, so genotyping stool pellets and seeing differences between the control mice and the malnourished mice. And actually one of the really important parts, I guess, about Plasmodium falciparum infection that I got to point out is it's associated with kidney damage, which is actually believed to be caused due to bacterial translocation. So we want to look at that as well. And with that, I'd like to make a few acknowledgements. I'd first of all like to thank my committee members, Dr. Michael Opata, Dr. Miriam Ahmed, and also Dr. Mary Kinkel, the animal facility manager, Monique Eckert, for helping take care of our mice, the microscopy facility manager, Dr. Geishwan Howell, for allowing me to use his microscopes. Lab members currently, Tyler Olander. Tyler, you've been a really important part in this research, and I'm really thankful for you. Past lab members, Amari Smith, Lindsay Richard, Jenny Pelotos, Mallory Reuter, Isaac Ogden, Margaret Smith, Annie Murray, and Kadra Ibrahim. And lastly, I'd also like to thank the Office of Student Research for funding this project and allowing me to do some really cool experiments and kind of paving the way for the future of the lab here. And lastly, and most excitedly, I also want to point out that this thesis data was actually accepted for publication and published on March 11th in a top 10% journal, so a Q1 journal with an impact factor of 4.5. So this is currently in the Journal of Nutrients. If you want to go and read that, that'd be great. And with that, I'd like to take any questions.
Noah, I have a quick question. So, in this, you were talking about how macrophages are part of innate immunity. They're some of the first responders to some of that bacterial dysbiosis that might happen with the gut integrity changing. Is there any reason that you looked at macrophages first and basically attributed some of this inflammation and that to those cells specifically?
So the reason why we looked at, well, we didn't even look at macrophages first. We looked at CD11B positive cells and also CD11C positive cells, which are early progenitors to both macrophages and dendritic cells. And the reason why we took the macrophages further and looked at those activation phenotypes was because that is more indicative, and there's more macrophages present in the intestines that can be causing inflammation versus other cell types. And I guess I think I kind of explained why we looked at interferon gamma was it's really prevalent for malaria infection and also gut barrier maintenance. So that's really the breakdown of why we looked at macrophages and their inflammation as well.
Okay, thanks so much. Great job.
Thank you.
That was a really great presentation, Noah. I had a question just about the kind of the baseline levels that you had in the macrophages in the gut. Was there any between the uninfected and the uninfected, both sides that were either malnourished or not? Was there a significant difference in the amount of macrophages in the malnourished versus the not malnourished? The reason why I ask is just because I wonder if there is like, because they're malnourished, there's already some, level of high inflammation there, maybe already starting from the macrophages and maybe from the leaky gut, and then that is just snowballing, if that makes sense. Is that kind of what you think might be happening, or is it you think that it's happening definitely after, you know what I'm saying? Does that make sense?
Yeah, so that's a really good question because so I first want to point out that our diet, it doesn't induce severe malnutrition. We consider it moderately malnourished. I think if we were to use a more severe example of malnutrition or even malnourishing these mice longer than 4 to 5 weeks, we could potentially see a difference in the absence of infection. However, based on the data you saw, we did not see any significant differences between the two diets without infection. We're suggesting here that while there was no difference, the effects of malnutrition are going to be exacerbated by malaria infection. So I think if we used a more severe model of malnutrition, we would potentially begin to see those differences, but that's kind of an open answer, I guess, to that.
Great, thank you.
Mm-hmm.
Any other question?
No, sorry, I was kicked out just when you were at the part that's most important, and you probably have answered this. I was wondering why the small intestine is more vulnerable. Is that because of macrophage? There's more macrophage in the small intestine?
So I really think there's two parts to that. I think, first of all, it's a much larger tissue, so there's more surface area, and so therefore more cells are going to be responsible for surveillance of microbial communities and sampling potential antigen. Secondly, there's also the Peyer's patches in the small intestine, which are lymphoid tissues. There's a lot of communication between the innate and immune response there. So I think that's the reason why we saw the greatest effect in the small intestine.
Okay, and so could you speculate? So in theory, then, if you were to supplement zinc to these animals, what would you expect to see?
Yeah, that's a great question, because that's actually one of our future directions, is to mimic the diet with just the absence of one micronutrient rather than the absence of zinc, iron, and deficiency in protein. So in regards to your question, I think reintroducing zinc back into the diet would probably help the situation and reduce the inflammatory environment, because like I said, zinc is really important for tight junction integrity. So under zinc deficient conditions, there's dysbiosis, which eventually causes inflammation. However, when you have enough dietary zinc, those tight junction proteins are well maintained. You're not going to have any dysbiosis and increases in inflammation.
All right, great presentation. Thank you.
Thank you.
Any other question?